Hello, everyone. Thank you for taking the time to listen to my presentation. My name is Shannon O'Connell. I'm an environmental studies student at the University of Oregon, and I'm in my senior year of college. And today I will be talking about my presentation titled Discovering the Genetic Blueprint for Zinc Enhanced Mandibles, where I got to work at the Schofield Lab under the guidance of my mentor, Robert Schofield. So, what does the Schofield Lab do? Well, as a brief introduction, the Schofield Lab emphasizes investigating the biophysics of ants, scorpions, and other invertebrates. This lab focuses on the peculiar materials leafcutter ants and many other invertebrates utilize for their teeth, claws, and other tools. These materials include zinc, manganese, and other unique elements. The element that I specifically emphasize on during my research project is zinc, which leads us to the research question. What genes are expressed in ant mandibles when zinc is included? Why do we care about this? Why is it important? Well, studying which genes are active in teeth with zinc will help us better understand the zinc biomaterial system. A zinc biomaterial system refers to how an organism uses zinc to strengthen parts of its body, such as teeth. This helps us better understand the role zinc plays in wear resistance in ant mandibles. To start this process, I must prepare ant mandibles and cut off the tooth strips to prepare for RNA extraction. I start this with step one. Collect a large leafcutter ant to ensure the mandible is large enough to easily cut. Once I select a large ant, such as the one in the first photo, I place him in a plastic tube and put him in the freezer to ensure a painless ethical death. Once the ant is ready, I take him out of the freezer and I begin step two, where I place the ant under a microscope and I attempt to remove the mandibles. And once I had successfully done that, I attempt to cut along the line of teeth. If you look at the second image below, you can see an image of the ant mandible with dotted lines, and that is roughly where I'm aiming to cut. After I have done that, I move on to step three, retrieve ant tooth strip, and I have to repeat the previous steps 8 more times. After that, I bring 8 tooth strips to the UO freezer at the genomics lab. And if you look at the third image below, you can observe the tooth strip that has been cut. That is roughly what we are aiming for. And it is important to know that in order to ensure the preservation of the ant tooth strips, I have to cut 8 of them in under 2 hours and bring them to the genomics lab freezer. So I have been practicing months, cutting off tooth strips, making sure that I can be fast enough to drop them off in the UO Genomics lab freezer. By gaining hands-on experience with leafcutter ants, I was able to learn about ant behavior and the importance of the materials that make up their mandibles. Despite struggling for months, I'm grateful for the opportunity to learn precise dissection skills. Unfortunately, the ant colony at the Schofield lab is declining. This factor decreases the number of ants to collect. With fewer large ants, the current project to extract RNA from ant mandibular teeth is halted. We are unable to fully collect results until we can collect sufficient data. In the meantime, I am learning to measure ant mandibles using the software ImageJ. Thank you so much for listening to my presentation and my work in progress as I learn to dissect ant mandibles. I would like to acknowledge and thank my mentor, Robert Schofield, for the guidance he had provided me during my time in his lab. And I'd also like to thank my lab partner, Elise, for training me to cut ant mandibles. Thank you so much, everyone, and have a great day.